disease mechanisms that need to be explored in ST. Functional neuroimaging studies are difficult to interpret in ST. However, some characteristics of the disorder suggest some potential neural bases for the disorder. First, onset is gradual, after which the disorder becomes chronic, suggesting some adaptation processes are involved in the development of the pathophysiology. Although only the speech production system is involved, speech is normal during whispering in ST and symptoms are dramatically reduced after botulinum toxin injection, suggesting that the speech production system is not permanently altered, as it can be normalized immediately with changes in laryngeal output. One interpretation could be that the increased cortical activation in the motor and sensory laryngeal cortices found on functional neuroimaging, Ali et al., 2006, Simonyan and Ludlow, 2010, may have developed in response to pathophysiology elsewhere in the neurolaryngeal control system and might represent compensatory mechanisms responding to interference with laryngeal muscle control downstream. The neuropathology findings in a single postmortem case, Simonyan et al., 2008, may indicate abnormalities affecting either the internal capsule affecting the corticobulbar pathway, the basal ganglia, or feedback loops that modulate cortical control. Perturbation studies in normal speakers have demonstrated that speech gestures respond rapidly to online changes in both articulator position, Abbs and Gracco, 1984, and auditory feedback, Larson et al., 2000, within 100 ms when there is a mismatch between the expected and altered feedback. Further, the lack of symptoms during whispering when the vocal folds are not vibrating suggests that changes in laryngeal sensory feedback either from the vocal fold mucosa or subglottal pressures in the trachea may play a role in the pathophysiology of the disorder. One hypothesis, then, might be that the pathophysiology may involve sensory feedback from the laryngeal periphery affecting cortical physiology in ST. Further research on the role of laryngeal sensory feedback in the manipulation of symptoms needs to be performed. The recent discovery of THAP, thymidose-associated protein, domain containing apoptosis-associated protein 1, THAP1, as being the basis for DIT6 dystonia, Bressman et al., 2009, Fuchs et al., 2009, has led to studies of THAP1 mutations in primary focal dystonias of early onset, Holden et al., 2010, and late onset, Zhou et al., 2010. Some studies have identified THAP1 mutations associated with generalized dystonia, which may first affect the larynx, Jarmati et al., 2009, while one study has reported on ST adult onset that did not progress to a generalized form of dystonia, Zhou et al., 2010. Possibly, a variety of mutations in THAP1 may play a role in the development of cervicocranial dystonias including ST, Ozelius and Bressman, 2010. However, the role of THAP1 mutations in ST is somewhat limited. Of 460 patients with ST screened for sequence variants in 3 exons of THAP1, only 1%, had mutations in THAP1. The ST patients with THAP1 mutations who had Dr. ST were female with a mean onset age of 57.8 years. 2 had single amino acid substitutions, P.F132S and P.A166T. As ST is often confused with other voice disorders, Ludlow et al., 2008, Roy et al., 2008, further study in well-documented ST patients with clear phenotype characterization is warranted. The putative transcriptional dysregulation produced by THAP1 needs to be determined for each of the coding mutations identified thus far in the THAP1 gene, with 9 coding mutations documented in one study on late-onset cases, Zhou et al., 2010, and 9 others in another study on early-onset dystonia, Holden et al., 2010. The functional consequences of these mutations in the mature nervous system need to be determined, and the possible mechanisms of neuronal disruption that could lead to abnormalities in the control of motor neuron firing need to be determined. Tamiya, 2009. The histopathology identified in the one postmortem case of spasmodic dysphonia raises questions about transcription dysregulation that could result in the accumulation of parenchymal clusters located close to the vessel walls. Simonyan et al., 2008. Such material, if found in other cases, needs to be studied to determine the proteomic composition. TAP1 models are limited to cell cycle pathways in humans, fish, and nematodes, and appear to be critical regulators of cell proliferation and cell cycle progression. Basir et al., 2008. TAP1 mutations need to be developed in mammalian models to examine the effects of these mutations on the mammalian laryngeal system. Attention should be given to self-limiting mechanisms for focal adult onset dystonias including ST. The mechanisms involved must differ from the neurodegenerative disorders such as Parkinson's disease and amyotrophic lateral sclerosis. Because no clear pathological inclusions such as Lewy bodies have yet been identified in focal dystonia, pathologic confirmation of the disease is not available and thus far only symptomatology is available for diagnosis. Research on possible basic mechanisms is clearly needed, and additional postmortem tissue amenable to proteomic analysis is a necessary first step. The possible role of TAP1 mutations needs to be explored. In conclusion, the level of knowledge of the pathologic mechanisms and the pathways involved in this and other focal dystonias is limited compared with progressive neurodegenerative disorders. As the disorder is not progressive yet results in a chronic disability, a different type of molecular mechanism is likely involved and needs to be determined.